Hi, this particular video is about GPCR proteins and also the notes provided by Brian K. Kobilka and Robert J. Lefkowitz for the studies of GPCRs, for which they were awarded the Nobel Prize in Chemistry in 2012. So, first of all, I will go over the basic use and function of what GPCRs help us to do. So, what we understand of GPCRs is that they are transmembrane receptors that facilitate signal transmission across the cell membrane. They have a characteristic long polypeptide chain that passes through the plasma membrane 7 times, hence, they're also called 7TM, both ends of which are capable of binding to particular molecules. The extracellular end has an N-terminus that can bind to ions or lipids or polypeptides, among other such agonistic or stimulatory biomolecules. The intracellular end has a C-terminus that can join with a particular protein composed of alpha, beta, and gamma subunits of guanine nucleotide. Hence, it is also called the G protein. Upon receptor to ligand binding in the extracellular space, the helical structure of GPCR causes a huge conformational change for the plasma membrane in the plasma membrane, hence increasing its affinity for G alpha subunit to bind with the GDP molecule, hence becoming active. Before this, it only had GDP. The G alpha was attached with GDP. Additionally, G-beta and gamma form a heterodimer that, just like G-alpha and GTP, can bind to certain effector molecules. This active state can further elicit downstream processes like binding of G-alpha with an enzyme like adenyl cyclase, or that produces CAMP nucleotide, which is CAMP, another secondary messenger. Notably, G-alpha and GTP bond is eventually hydrolyzed by other chemical processes like RGS binding, which is the regulator of G protein signaling, and hence it returns back to the inactive state. This inactive state is described as GPCR bound to the trimer G-alpha, beta, and gamma, with the G-alpha bound to GDP molecule. And as you can see in this particular cycle, everything is represented exactly like that. On top of this knowledge, we also are aware of certain things, such as every human cell receives external stimuli and it affects the internal machinery through biochemical activities. So the basic function that GPCRs achieve is that they provide a framework to transmit information across the plasma membrane. So this is a ligand-activated receptor, and the research on it has been ongoing since 40 years. Now, early experiments had measured response of simulators or agonists, and even inhibitors or antagonists. And what laid the foundation of this research was the classical receptor theory, which is basically related to the mass action and the dose to response data of the drugs. So this entire GPCR is composed of 4 signaling components: the enzyme adenylyl cyclase, the CAMP-dependent kinase, heterodimeric G proteins, and CAMP with CAMP. These are the signaling components, and CAMP itself is an aftermath of a Nobel Prize. So Lefkowitz proved that receptor binding through radioactive labeled agonist ligands is possible, and he also had multiple researches based on epinephrine. So epinephrine is a neurotransmitter which is responsible for psychological processes such as fight or flight response. Also, in simple terms, it is the regulation of heart rate and even blood pressure. So cells with adrenergic receptors have 9 different forms, grouped as alpha and beta receptors. Some computational models and thermodynamic coupling shows interaction between ligands and G protein binding events, and this formed the ternary complex model. The agonist, which can be a hormone, neurotransmitter, odorant, etc., binds to the extracellular part of the receptor, while the G protein binds to the intracellular region, mutually contributing to the affinity of each other. Now, the gene for GPCR has no introns. Kobilka and Lefkowitz sequenced it in entirety and found similarity between rhodopsin receptor and beta AR, which is the epinephrine receptor. So this concluded that all GPCRs might have 7 transmembrane protein and may span across the plasma membrane. Now, in the signaling method, the ligand does not actually pass to the receptor. It only activates further processes by conformational change, hence increasing the ligand's affinity to bind to the receptor. So the ligand binding causes the receptor to increase the affinity of G protein inside the cell as well, which causes the G protein to dissociate into G alpha, beta, and gamma. And only the G alpha associates to the receptor and simulates enzyme like adenylyl cyclase, which produces CAMP nucleotide, and it serves as a secondary messenger, as I've already mentioned before. There are other proteins that may bind to G, beta, and gamma to modulate further signaling. One agonist molecule may lead to many G protein activating, and hence it amplifies a signal like that, since this complex is short-lived. Then the nucleotide hydrolysis causes the G protein to reassociate as the ligand is flushed away. This is when the ligand to receptor binding is closed. Now, the helical shape of GPCR, which had earlier caused a big conformational change, returns to its normal resting stage. And here is where you can still see it using techniques like NMR, IR, or fluorescence. The intracellular regulation via kinases like adrenergic or rhodopsin kinase increases the receptor affinity for arrestins that leads to a downregulation of the 7 transmembrane proteins function. So this indicates that the 7TM receptor can be desensitized to ligand binding externally as well. I hope these notes were helpful. Thank you for watching.